In this lecture, we are going to discuss various cancers as well as the molecular methodologies to diagnose these cancers. At the end of the lecture, you should be able to compare and contrast tissue-specific and tumor-specific molecular targets, list molecular targets that are useful for diagnosis and monitoring of solid tumors, explain how microsatellite instability is detected, show how clonality is detected using antibody and T-cell receptor gene rearrangements, describe translocations associated with hematological malignancies that can be used for molecular testing, identify genetic abnormalities associated with various cancers, and define several cancer-related terms such as oncogene, loss of heterozygosity, and allelic replacement. So oncology is a study of tumors, and a tumor or a neoplasm is a growth of tissue that exceeds and is not coordinated with normal tissue. A benign tumor is a tumor that is not recurrent. A malignant tumor is invasive and tends to recur at multiple sites. And cancer is a term that includes all malignant tumors. In general, cancer is divided into two broad groups: solid tumors and hematological malignancies. For solid tumors, these are designated according to their tissue of origin. So, for example, carcinomas are tumors that originate in the epithelium. Sarcomas are tumors that originate in bone, cartilage, muscle, blood vessels, or fat, and teratocarcinomas are of multiple cell types. Metastasis is the movement of dislodged tumor cells from the original or primary site to other locations, and only malignant tumors become metastatic. And hematological malignancies, these are tumors that arise from white blood cells. And the two main hematological malignancies are leukemia and lymphoma. Leukemia is a neoplastic disease of blood-forming tissue where large numbers of white blood cells populate the bone marrow and peripheral blood. A lymphoma is a neoplasm of lymphocytes that forms discrete tissue masses. Lymphomas are further divided into Hodgkin's disease or plasma cell neoplasms. And Hodgkin's disease is a histologically and clinically different disease than all other types of lymphoma, which are called non-Hodgkin's lymphoma. Plasma cell neoplasms arise from terminally differentiated B cells and are classified in a separate category. Cancer is caused by non-lethal genetic mutations in DNA affecting certain genes. And the genes that are commonly affected, which lead to cancer, are oncogenes and tumor suppressor genes. So an oncogene, which functions as a proto-oncogene, usually promotes cell division or cell survival. If you have a mutation in the oncogene, this usually results in a gain of function, and these mutations are dominant. There have been more than 100 oncogenes identified in the human genome. Tumor suppressor genes normally arrest cell division, and mutations in tumor suppressor genes usually usually cause a loss of function and are recessive mutations. So this is a schematic of your non-lethal genetic mutations. So you have mitogens, which are proteins that are necessary for your normal cell growth. If you have a mutation without any mitogen, you are going to usually kill that cell. You don't want mitosis to occur. So you're going to arrest the cell growth, and possibly the cell will die through apoptosis or programmed cell death. If you don't have your mitogen, and you have an oncogene or a tumor suppressor gene mutation, then you're going to get abnormal or uncontrolled cell growth. Targets to detect cancers. There are two types, tissue-specific markers and tumor-specific markers. Those are the two main types. And then there are also viruses that are associated with causing several different types of cancers. So your tissue-specific markers are molecular characteristics of the type of tissue from which a tumor arose. The presence of DNA or RNA from these targets in abnormal amounts or in locations that you wouldn't normally find the DNA or RNA are used to detect and monitor cancers. These are usually going to be antigens or gene rearrangements. And although the tissue-specific markers or targets are useful, they are also expressed by normal cells, so they don't always prove that cancer is present. Tumor-specific markers are not expressed in normal cells, and these are very indicative that a cancer is present. And these usually you're going to be looking for translocations, point mutations, polymorphisms in tumor suppressor genes or oncogenes. Also, since some viruses are associated with cancer, you can look at viral detection, such as Epstein-Barr virus, hepatitis C virus, HTLV1, and there's several other viruses that have been associated with cancer. You're going to use your standard molecular methods to detect these specific markers or targets, such as hybridization and blotting, your standard polymerase chain reaction, reverse transcriptase PCR, electrophoresis, other types of PCR, such as heteroduplex analysis, and single-strand conformational polymorphism. Also, you can use real-time PCR with either a gene-specific probe or a patient-specific probe. Your gene and chromosome abnormalities that are observed in cancer. So we already said you get mutations in genes, and usually these are going to be your oncogenes or your tumor suppressor genes. You're going to see various chromosomal structural abnormalities like translocations, deletions, and insertions, as well as abnormal chromosome numbers such as aneuploidy and polysomy. So the first target that we're going to talk about is the human epidermal growth factor receptor 2. This is also called HER2/neu. This gene is located on chromosome 17, and it encodes one of a family of human epidermal growth factor receptors. So this gene is frequently amplified in breast cancer cells, and it results in increased amounts of the HER2 protein on the cell surface. HER2/neu overexpression is a predictor of a more aggressive growth, breast cancer growth, and also metastasis. HER2 expressing tumors are sensitive to a monoclonal antibody therapy called Herceptin. The HER2 protein is detected by immunohistochemistry, and HER2/neu gene amplification is detected through fluorescence in situ hybridization, or FISH. So several factors such as epidermal growth factor, transforming growth factor alpha, neuregulins, and hairregulins bind to these receptors on the cell surface and begin a cascade of events, including autophosphorylation and phosphorylation of other proteins by the receptors. So epidermal growth factor receptor, or EGFR, is located on chromosome 7, and this is an oncogene that encodes another of the same family of epidermal growth factor receptors. So the EGFR gene is mutated or amplified in several types of cancer cells, and overexpression of EGFR has been correlated with poor clinical outcome. Tumors with activating mutations in EGFR are sensitive to tyrosine kinase inhibitors. The EGFR protein is detected by immunohistochemistry, and EGFR gene and chromosome abnormalities are detected by FISH. Gene mutations are detected by single-strand conformational polymorphism, as well as other types of PCR and sequencing. The G protein family members, or the RAS genes, these are the Kirsten-Rat sarcoma viral oncogene homolog, or KRAS, which is located on chromosome 12, neuroblastoma RAS, NRAS, located on chromosome 1, and Harvey-Rat sarcoma viral oncogene homolog, or HRAS, located on chromosome 11. So the mutations in these RAS proto-oncogenes occur in many different types of cancers. These three RAS genes in mammals produce four very similar proteins: KRAS4A, KRAS4B, NRAS, and HRAS, which differ only in their carboxyl terminus. These RAS proteins bind and hydrolyze GTP for energy. KRAS encodes a key component of cell signaling. Mutations in KRAS are the most common oncogene mutations in cancer. The most frequently occurring mutations are located on codons 12, 13, 22, and 61 of the RAS gene. Mutations throw the RAS protein into a permanently active state. These mutations are associated with tumor malignancy and may affect responses to some therapies. KRAS gene mutations are usually detected by SSCP or direct sequencing. Ewing's sarcoma. The Ewing sarcoma gene is located on chromosome 22. And this is a group of tumors arising from primitive neuroectodermal tissue, or PNET. So the Ewing sarcomas comprise a family of childhood neoplasms. There are no unique characteristics that distinguish the different types of tumors that make up this Ewing sarcoma group. Diagnosis is aided by the detection of HbA7-1 cell surface markers through immunohistochemistry staining, and detection of specific translocations can be used for diagnostics and prognosis. The Ewing sarcoma translocations, there's several that are much more common. So translocations involving the EWS gene at chromosome 22 with the FLI1 gene at chromosome 11 are present in 85% of cases. Translocation between the Ewing sarcoma gene and the ERG gene, that this is present in 5 to 10% of the cases, and there are other translocation partners for the Ewing sarcoma gene, and all of these other translocations are present in fewer than 1% of cases. So molecular testing we can do for Ewing sarcoma. We can do reverse transcriptase PCR, and when doing this, we would reveal a 120 to 190 base pair amplicon, or PCR product, whereas in negative specimens you will get no product at all. Now, considering that this specific test gives you no product at all, you really need your amplification controls here. So you need to use your constitutively expressed genes such as GAPDH or 18S RNA, so that you know if these amplify, then everything about your PCR was perfect and where you didn't yield a product that's a true negative as opposed to a false negative. Here's an example of the Ewing sarcoma reverse transcriptase PCR detection, where you have a translocation between the EWS gene at chromosome 22 and the FLI1 gene at chromosome 11. You get a fusion of these two genes. When you extract your messenger RNA for your RT-PCR and then convert that into complementary DNA, you're going to use a primer, a forward primer that's specific for the EWS gene, and a reverse primer that's specific for the FLI1 gene. You are only going to get a product, you're only going to amplify if this translocation is present. Synovial sarcoma is a rare type of cancer of the muscle, fat, fibrous tissue, blood vessels, and other supporting tissues. It accounts for 8 to 10% of all sarcomas and is more common in young adults. 80% of synovial sarcoma is caused by a recurrent reciprocal translocation between chromosome 18 and the X chromosome. The translocation fuses the synovial sarcoma in chromosome 18 with either of two related genes on the X chromosome. So you get the synovial sarcoma gene, which is SS18 or SYT, and that is going to cause a fusion with either SSX1 or SSX2 genes on the X chromosome. You can detect these translocations through FISH or reverse transcriptase PCR. Rhabdomyosarcoma is the most common soft tissue sarcoma of childhood. It accounts for 10% of all solid tumors in children, and there are three forms: alveolar rhabdosarcoma or ARMS, embryonal rhabdosarcoma, and primitive rhabdosarcoma. The alveolar rhabdosarcoma has a worse prognosis. Translocations in the fork head of the rhabdomyosarcoma gene, or FKHR, which is also called FOXO1A, with the paired box genes PAX3 and PAX7 are more common in the alveolar rhabdosarcoma. Tumor protein 53 or TP53, this is a 53 kilodalton tumor suppressor gene that encodes a transcription factor. TP53 is mutated in half of all types of cancers. Loss of TP53 function is an indicator of poor prognosis in colon, lung, breast, and other cancers. Mutant p53 protein is detected through immunohistochemistry, and TP53 gene mutations are detected through various methods, including sequencing and SSCP. The ataxia-telangiectasia mutation gene, we discussed ataxia-telangiectasia a while back in one of the first several lectures. This is a neurological disease that causes a predisposition to cancer. It occurs in 1 to 40,000 live births, and it's caused by mutations in the ataxia-telangiectasia mutation gene, the AT mutated, and this is a gene on chromosome 11. So ataxia-telangiectasia mutated gene mutations also occur in some types of leukemias and lymphomas, and carriers for the autosomal recessive mutation in ATM are at increased risk for developing leukemia, lymphoma, and other cancers. So diagnosis of ataxia-telangiectasia, you can do direct sequencing of the ATM gene as well as SSCP to look for mutations. And there's also a functional test that we discussed previously to look for repair of double-strand breaks that are induced by radiation. So what you would do is you would take your exponentially growing cells and irradiate them. After 2 hours, you add Coulsomid to inhibit spindle formation. The cells are harvested 2 hours later, and Giem sustaining is done in examination of the karyotype. Then you do a ratio of aberrations per cells, and this is calculated from the number of chromatid and chromosome breaks, as well as other abnormalities in the chromosome structure. The breast cancer 1 and 2 genes are BRCA1 and BRCA2. BRCA1 is located on chromosome 17 and BRCA2 is located on chromosome 13. 5% of breast cancers result from inherited gene mutations in these two genes especially. Women with a mutation in the BRCA1 gene have a 60 to 80% lifetime risk of breast or ovarian cancers. Men carrying mutations, especially in the BRCA2 gene, have a 100-fold increased risk of breast, colon, and prostate cancer. There are 3 specific mutations that are more common among certain ethnic populations. So you have a deletion, or 2 different deletions, and an insertion. 2 of these, a deletion and an insertion in the BRCA1 gene, and 1 deletion in the BRCA2 gene. So BRCA1 and BRCA2 are tumor suppressor genes that encode proteins that participate in DNA repair. So we already said inherited mutations in either of these genes increases a person's risk of breast cancer at an earlier age, and frequently occurring mutations are detected by SSP, PCR, as well as other methods. Most mutations are detected by direct sequencing of both the BRCA1 and BRCA2 genes to identify mutations. So if you were to detect the BRCA1 deletion by SSP PCR, you have a mutation in your BRCA1, your deletion. You would use a mutation-specific primer. You would also have other primers that are going to amplify different regions. So you have a forward primer, and your mutation-specific primer are going to amplify a 180 base pair amplicon. You also have another forward primer in front of the mutation, and another reverse primer, and that forward and reverse primer are going to give you a 120 base pair amplicon. So then you would run your gel after you did your PCR. You have your molecular weight standard. You have your positive lane, that's a normal. You have the M lane, that's a mutant, and your B is the blank. So the very first lane is your molecular weight. The second lane is a normal individual that does not have the 180 base pair band. The third lane is a mutant, and you can see the full product from the forward and the far right reverse primer is 230 base pairs. You also have your 180 base pair band with your mutation-specific reverse primer, and you also have your 120 base pair band. So that's what you're looking for with this specific PCR reaction. Von Hippel-Lindau gene, or VHL, is located on chromosome 3, and it's a genetic condition involved in abnormal growth of blood vessels in organs, and especially the organs that are rich in blood vessels. It's caused by mutations in the VHL gene, and VHL syndrome is a predisposition for renal cell carcinoma and other cancers. Deletions, point mutations, and splice site mutations have been described in VHL patients. The NMYC gene, well, these MYC genes, there's VMYC avian myelocytomatosis viral related oncogene and neuroblastoma derived MYCN or NMYC located on chromosome 2. These are amplified in cases of neuroblastoma and retinoblastoma. The NMYC is an oncogene that is counteracted by the tumor suppressor gene neurofibromatosis type 1 or NF1. Rearranged during transfection gene or RET is a proto-oncogene located on the long arm of chromosome 10. The RET or rearranged during transfection gene is an example of how different mutations in the same gene result in different diseases. So translocations that result in overexpression of your rearranged during transfection are found in thyroid papillary carcinomas. Point mutations that activate this gene, the RET or RET, is also called MEN2A. Point mutations that activate this gene are found in inherited multiple endocrine neoplasia syndromes, so multiple endocrine neoplasia, MEN syndromes. Loss of function mutations in this gene are found in Hirschsprung's disease, which is a rare congenital disorder resulting in colon obstruction. Hereditary non-polyposis colorectal carcinoma, or HNPCC, also known as Lynch syndrome, accounts for about 5% of colon cancer. So hereditary non-polyposis colorectal carcinoma, or HNPCC, is the most common form of hereditary colon cancer and is associated with mutations in genes encoding components of the mismatch repair, or MMR system, most frequently the MLH1 and MSH2. We had already discussed previously that you can get errors during replication. And microsatellites, or our short tandem repeats, are very sensitive to these types of replication errors. Now, the mismatch repair system is a repair system in your cells that are going to correct these errors. Components of the mismatch repair system are encoded by that MLH1, MSH2, and several other genes. Microsatellite instability is the production of new alleles from unrepaired replication errors. So if you have a problem in your mismatch repair system, you're not going to repair these commonly occurring errors during replication. So then you're going to produce new alleles that never were repaired through the mismatch repair system, which is microsatellite instability. So here's a schematic where you have, in this region, you have your short tandem repeat, you have 7 T's. So on the opposite strand, you're going to have 7 A's. Well, if all of a sudden you end up with, on one strand, only 6 T's, then you're going to end up with getting a mismatch, and normally your mismatch repair system is going to correct that, but occasionally that doesn't happen. So you would end up getting a new allele that's generated on the next round of replication. So how is this microsatellite instability related to the HNPCC? So 85 to 90% of the HNPCC tumors have microsatellite instability. Mutations in genes of the mismatch repair system, these are loss of function mutations, are inferred by testing for microsatellite instability. So microsatellite instability analysis determines the gene function, and direct sequencing is used to detect the actual gene mutation. So microsatellite instability is analyzed by assessing the stability of at least 5 microsatellite loci, and these 5 loci are what's recommended by the National Cancer Institute. So you have 5 different markers, 2 of which are mononucleotide repeats and 3 are dinucleotide repeats. Inherited tumor suppressor gene mutations are recessive for the malignant phenotype. Tumor suppressor gene mutations are dominant with respect to increased risk of malignancy, and loss of heterozygosity reveals the loss of the good allele at a locus, which uncovers the homologous locus with a recessive mutation. So this phenomenon is explained by the two-hit hypothesis. So usually, in general, the two-hit hypothesis is you need both of your alleles to become mutated before you get cancer. So normal, you have 2 normal alleles. You're considered at risk if you have a mutation in one allele, but your other allele is normal. If that occurs, usually you're not going to get cancer. However, if both alleles have the mutation, that's when you are going to end up getting cancer. That would be considered you're affected. Now, if you have inherited mutation in one allele, that means that if you inherited a mutation in one, you only need one mutation, not 2, to get cancer. So if you generate a mutation in your good allele, you could end up getting cancer. Or what happens is you lose the good allele, your loss of heterozygosity. So then you only have the defective allele, which can then generate cancer. So your loss of heterozygosity can be detected through short tandem repeat analysis. So you have a loss of a linked heterozygous short tandem repeat, which implicates a concurrent loss of one gene allele. So on the left you have your schematic where you have your 2 alleles. One contains a mutation and one is normal. And in this instance, if you did capillary gel electrophoresis, you'd have 2 peaks, one for your normal allele and one for your mutant allele. But if you have a loss of heterozygosity, you're going to keep your allele with the mutation and you're going to lose the normal allele. So when you do your capillary gel electrophoresis, you're going to get a peak for your mutant allele, and for your normal allele, you're either going to not get a peak or you're going to get a very reduced size of the peak. Microsatellite instability is detected by comparing PCR amplicons of the microsatellite loci. Unstable loci appear as extra products in the tumor tissue compared to normal tissue. So you can look at a normal agarose gel, or you can do your capillary gel electrophoresis. So in a normal agarose gel, if you compare normal tissue to tumor tissue in your unstable loci, you're going to get a lot more bands in the tumor compared to normal. Your stable loci will look the same. Same thing with capillary gel electrophoresis. In your unstable loci, you're going to get numerous more peaks in your tumor tissue compared to normal. Whereas if you have a stable locus, you're going to get the same peak size and pattern. Detecting leukemia and lymphoma using molecular techniques, you're going to look at targets such as antibodies, gene rearrangements, translocations, point mutations, polymorphisms, as well as viruses. And the methodology is going to be the same: hybridization, PCR, reverse transcriptase PCR, real-time PCR. So gene rearrangements, these are normal events that occur in lymphocytes. Antibody genes, such as the immunoglobulin heavy chain genes, the immunoglobulin light chain genes, and the T cell receptor genes, rearrange, which contributes to antibody diversity. Rearrangement occurs independently in each cell, and the completion of gene rearrangement on only one of the two immunoglobulin heavy chain gene alleles is referred to as allelic exclusion. So here's a schematic of your immunoglobulin and T cell receptor gene rearrangements, where you're beginning with your lymphoid stem cells. You get your early cells, and then you get your mature plasma cells, cytotoxic T cells, and helper T cells. And rearrangements will begin early in the process of maturation. So immunoglobulin heavy chain rearrangement, or what's known as VDJ rearrangement. So you have in your immunoglobulin heavy chain, you have your variable regions, which are your VH regions, and these are preceded by a leader region. You have your diversity region, or your DH region, and your joining regions, which are your JH regions. And one of each gene segment is selected and joined, and the intervening DNA is then looped out. And then you get your introns, which are spliced out, and you get your V, your D, and your J that are going to rearrange. So your immunoglobulin light chain genes and T receptor genes rearrange in a very similar manner to the immunoglobulin heavy chain genes. Gene rearrangement may be used to detect leukemias and lymphomas arising from cells that have rearranged their immunoglobulin or T cell receptor genes. Normal lymphocyte populations are polyclonal with respect to their immunoglobulin and T cell receptor genes. However, a leukemia or lymphoma is monoclonal with regard to the immunoglobulin or T receptor rearranged genes. So whenever you have over 1% of the cells that make up the same gene rearrangement, that cell population is referred to as monoclonal, because each cell rearranges individually. So normally you're going to have polyclonal rearrangements. Each cell is going to vary. Whenever you have a larger percentage of the cells that all have the exact same gene rearrangement, this is your monoclonal, and this usually is indicative of a leukemia or a lymphoma. So you can detect these monoclonal lymphocyte populations through southern blotting. So what happens is, as you know, to do your southern blot, you take your DNA and you digest it with restriction endonucleases. Usually you're going to get a certain pattern on a gel or a fingerprint, a banding pattern. And when you get mutations, you're going to either add or remove or change restriction sites. So you're going to get a variation in the banding pattern when you cut the DNA with your restriction enzymes. So here's an example of your region of DNA and your cut sites. In this example, you're using BAMH1, HND3, and EcoR1. When you take your DNA, you digest it with your various enzymes, you run it on a gel, you transfer it to nitrocellulose membrane, and then you are going to blot the membrane with a probe that is labeled so that you can detect it. If the probe finds a homologous sequence, it is going to hybridize to that region, and that's going to show up as a band. So if you look at EcoR1 digested DNA, BAMH1 digested DNA, and HND3 digested DNA, comparing your germline cells to your rearranged cells, if you get an extra band or several more bands in your rearranged cells compared to your germline cells, that would be indicative of a positive or a monoclonal population, which indicates a leukemia or a lymphoma. You can also use polymerase chain reaction. And what's going to happen is your monoclonal populations will be detected through a sharp band that's unique to the tumor cell population. So again, you have your region of DNA, you have your primers, and you're going to amplify various products depending on what population of cells you have. So normal populations or polyclonal populations are going to give you a polyclonal PCR product. And since there's so many amplicons that are being made, you don't get really nice distinct bands. You get a very diffuse and dispersed banding pattern. This is indicated in lanes 4, lanes 8, and lanes 11. You can see you can't really see nice sharp crisp bands there. However, if you have a monoclonal population, you are going to get a very sharp band and a single PCR product. And you could see this in lanes 3, 6, 9, and 10. That would be indicative of a monoclonal population. So gene rearrangements in your immunoglobulin and T cell receptor genes are normal. And you're going to get these rearrangements regardless of the fact that a malignancy is present. Targeting clonality of gene rearrangements can only be applied to tumors arising from lymphocytes. If you have other malignancies, you have to use other targets for diagnosis, such as translocations and other mutations. So here are some of the examples of the common translocations that can be used to diagnose hematological tumors. So there's various types of tumors there: B-cell leukemia, acute myelogenous leukemia, etc. Here are more translocations for hematological tumors. So translocations and other abnormalities in the chromosome structure or number are usually detected through fluorescent in situ hybridization. So you can use your fluorescently labeled probes and look at your chromosomes through your FISH. So your translocation detection can be done using FISH breakaway probes. So here you have your chromosome 14 and chromosome 8, and in the schematic on the right, you can see that a translocation has occurred between your chromosome 8 and 14. Translocations are detected with higher sensitivity using amplification, such as polymerase chain reaction. Quantitative PCR may be used to quantify tumor load during patient monitoring. Fluorescent in situ hybridization is recommended for initial diagnosis, and PCR is a better method used for monitoring. A reciprocal translocation between the long arms of chromosome 14 and 18 is found in 90% of follicular lymphoma cases and 20 to 30% of large cell lymphomas. With this translocation, the B cell leukemia and lymphoma BCL2 gene is moved from chromosome 18 to chromosome 14. BCL2 is dysregulated and overexpressed when it's moved from 18 to chromosome 14. A breakpoint is where one chromosome has split and the DNA is rearranged by the insertion of a piece from another chromosome or a different part of the same chromosome. There are several breakpoints in chromosome 18 region 3 to the BCL2 gene, most which occur in the major breakpoint region or MBR. 10 to 20% of breakpoints fall into a cluster closer to the BCL2 gene in the minor cluster region or MCR. An intermediate cluster region and other breakpoints also exist. So using polymerase chain reaction to detect the translocation between chromosomes 14 and 18, you use a forward primer which hybridizes to chromosome 18 and a reverse primer which hybridizes to chromosome 14. And the band size that you get, your amplicons, are going to be determined by the chromosomal breakpoints. So you can use a variety of primers. You could use primers specific for the major breakpoint region, the minor cluster region. You always have to have your molecular weight marker. If you look at your gel, the positive lanes are positive for a translocation and the negative lanes are negative for a translocation. So you could see in your positive lanes, you see nice bright bands, and your negative lanes, you don't really get amplification. A reciprocal translocation between the long arms of chromosome 9 and 22 is found in chronic myelogenous leukemia and acute lymphoblastic leukemia. This specific translocation forms a chimeric gene between the breakpoint cluster region gene on chromosome 22 and the Abelson leukemia virus gene on chromosome 9. The translocated chromosome is the Philadelphia chromosome. So the chimeric gene, BCR-ABL, produces an abnormal protein that drives the tumor cell phenotype. So you could see your translocation where you get your fusion of the two different genes, which is going to make a fusion protein. You can detect these translocations using reverse transcriptase PCR. So this specific translocation between 9 and 22, you have your BCR gene, your ABL gene, the Philadelphia chromosome. Those are spliced together. You get your messenger RNA, which is then reverse transcribed into complementary DNA, and then you could use your PCR with primers specific, one forward primer to the BCR, the reverse primer to the ABL gene, and if you get a product, you know that you have that translocation and fusion. So in this gel, you can see lane 1 is the molecular weight standard, lanes 2 to 5 are positive for translocation, 6 is negative, 7 through 11 are your amplification controls. So we said previously that whenever you have these amplification reactions where no amplicon at all is considered negative, you really need to make sure you have all of the appropriate controls present, especially your amplification controls, so that you know that your reaction worked properly and that if you don't have a band, it's because it's a true negative. Lane 12 is a blank. And you could see your nice band, and the band size is determined by different chromosome 22 breakpoints. You can also quantify using qPCR, the quantitative PCR or real-time PCR TACMAN procedure. So for qPCR, you can use a standard curve of tumor cells that's diluted into normal cells. For real-time qPCR, you can use a standard curve of transcripts of known copy numbers that's diluted into normal RNA. So, in summary, molecular testing analyzes tissue specific and tumor specific targets or mutations. Genome, chromosome, and gene mutations are targets for diagnosis and detection of solid tumors. Microsatellite instability is a test for function of the DNA mismatch repair system, which may be mutated in hereditary colon cancer. Immunoglobulin and T cell receptor gene rearrangements are tissue specific markers for certain lymphomas and leukemias. Translocations are tumor specific markers for some hematological disorders.